Hey guys, welcome to today's lesson, Chapter 12, Section 3, which is all about DNA, RNA, and protein. So we've already talked about important biomolecules of life in the past. Protein was one of them. And if you look at this statement here, it says DNA codes for RNA, which guides protein synthesis. So basically, DNA makes RNA, which causes protein to be created. So think about protein, think about what you already know about protein, and I want you to take a whole minute to think about why is protein important for us. Go ahead and pause the video and think about that for 1 minute and then come back. All right, well, hopefully you had a chance to think about that, and we are going to look into how DNA codes for RNA, which guides protein synthesis. And we know protein is important for us. Today's essential questions are: how are messenger RNA, ribosomal RNA, and transfer RNA involved in the transcription and translation of genes? What is the role of DNA polymerase in the synthesis of messenger RNA? And how is the code of DNA translated into messenger RNA and utilized to synthesize a protein? Well, first, let's talk about what we call the central dogma. After the discovery of DNA's structure, which is that double helix shape that we talked about in the past, scientists turned to investigating how DNA served as a genetic code for the synthesis of proteins. Geneticists accept that the basic mechanism for reading and expressing genes is from DNA to RNA to protein. This is referred to as the central dogma of biology. DNA codes for RNA, which guides the synthesis of proteins. So because we have DNA, and it has all the information to make RNA, and then RNA has all the information to make proteins, essentially DNA leads to our protein, which is a pretty important thing. So RNA, we know what DNA is, we've talked about that, but RNA is very similar. It's a nucleic acid that's very similar to DNA, but it's made with the sugar ribose, not deoxyribose. That's why it's RNA instead of DNA. And it also has what's called uracil instead of thymine. So instead of a T, we have a U. There are 3 types of RNA. We have messenger RNA, ribosomal RNA, and transfer RNA. So messenger RNA, or mRNA, these are long strands of RNA that are formed complementary to one strand of DNA. Direct synthesis of a specific protein comes from this kind of RNA, mRNA. Now, when it says it's a long strand of RNA that's formed complementary to one strand of DNA, that means as it's created, it makes the complementary type. If you have adenine, it's going to match up or create uracil instead of thymine in this case. Or if you have guanine, it's going to match up and make cytosine. So think back to how those pairs were complementary. This is going to make a complementary strand. So the RNA is complementary to the DNA that it was copied out of. Now, ribosomal RNA, or rRNA, associates with the proteins to form ribosomes in the cytoplasm. So rRNA basically makes the ribosomes in the cytoplasm. And then transfer RNA, or tRNA, these are smaller segments of RNA that transport amino acids to the ribosomes. Okay, for RNA to form, we have what's called transcription and translation. So first let's look at transcription. The first step of the central dogma involves the synthesis of mRNA from DNA in a process that we call transcription. So the enzyme called RNA polymerase regulates RNA synthesis by binding to the specific section where an mRNA will be synthesized. So it's going to attach onto a part of DNA that's going to start creating RNA. And RNA polymerase moves along the DNA strand in a 3' to 5' direction, and as it moves, it synthesizes a strand of mRNA. It has a brand new strand out of the old strand of DNA. And you can see in this picture, this RNA polymerase as it runs across here, it's going this direction. If you follow my mouse here, if there's a C on the DNA, it makes a G on the RNA. If it's a G on the DNA, it makes a C on the RNA. A to U instead of T. T goes to A. C to G, A to U, A to U, T to A, T to A, and so forth. It's just like when we talked about the complementary strands last time around. It makes complementary strands of RNA. Okay, RNA processing. When we compared DNA code with its transcribed mRNA code, scientists found that the mRNA code was much shorter. So DNA sequences not found in mRNA are called introns, and DNA sequences that remain in the final mRNA are called exons. So the pieces that are kind of snipped out are the introns, and the pieces that kind of stick around and get combined to stick around in the final mRNA, those are exons. Now, there's a specific code for every three sets of bases, there's a code. And we're going to look at that for here in a second. But scientists hypothesized that the instructions from protein synthesis were encoded in the DNA. And experiments during the 1960s demonstrated that the DNA code was a three-base code. The three-base code in DNA or mRNA is called a codon. So these little 1, 2, 3, 1, 2, 3, these little three-base sets is called a codon. And each codon codes for what we call an amino acid. And those amino acids eventually lead to proteins. We'll get into that in a second. Okay, so we've had transcription. We get our new strands of mRNA from our DNA. Well, next we have what's called translation. After the synthesis of mRNA, the mRNA moves from the nucleus into the cytoplasm, where it connects at the 5' end to a ribosome. The mRNA code is read and then translated into a protein through a process called translation. So if you look at this picture here, we have our three base pairs or our codon, and we have several codons. And what's called an anticodon, the complementary opposites of the codon, and those anticodons come in to make a complementary strand, which leads to new proteins. So tRNA molecules act as the interpreters of the mRNA codon sequence. The tRNA is activated by an enzyme that attaches at a specific amino acid to the 3' end. The middle of the folded tRNA contains an anticodon, which is a complementary sequence to the mRNA codon. That's what we said was this right here in the picture. An anticodon is complementary to our codons. Okay, now the role of the ribosome. So ribosomes provide a site for protein synthesis. When mRNA leaves the nucleus, the two ribosomal subunits come together to hold the mRNA in place for translation. The ribosome structure has grooves that hold on and serve as tRNA sites for amino acid attachment. Now, there was an experiment called the Beadle and Tatum experiment, which showed that one gene codes for one enzyme. And we now know that one gene codes for one polypeptide. And that is the end of this lesson. If you need to go back and review transcription and translation, I would recommend doing that to make sure you have the parts down, what goes on during transcription, what goes on during translation, know the difference. And once you're all finished reviewing, make your way over to this week's discussion board.